We will continue our discussion on plasma membrane, and in this particular video we will be talking about membrane transport, and specifically today we are targeting passive transport across plasma membrane. As we already know, the gases like oxygen or carbon dioxide can readily enter or leave our cells through plasma membrane. We say these gases or these gaseous molecules are readily permeable through a plasma membrane. But when it comes to large molecules like sugars, like glucose cannot enter cells on its own. Similar thing happens with very small charged ions like sodium ion, like potassium ion, magnesium, calcium, chloride. All these ions can simply not enter our cells through plasma membrane. So, what do we actually mean by the permeability of membrane? So, the permeability of membrane is defined with respect to rate of diffusion. How many molecules of oxygen are entering in cell through this plasma membrane in a particular amount of time. For an example, we are considering that approximately 10,000 molecules of oxygen are entering our cell through this plasma membrane in 1 second. So, we take this as a standard for permeability. Alright, but when it comes to large molecules like glucose, fructose, this number goes way below 1000 molecules per second. So, such a small number, we can say that this membrane is relatively impermeable to glucose and fructose. Similar thing happens with these small charged ions like potassium. This number goes way below 500 molecules or 500 ions per second. So, we can say that this membrane is relatively impermeable to these small charged ions. And when we combine these two factors like large molecules and charge, we get charged polar molecules like amino acids like adenosine triphosphate, glucose 6 phosphate, various nucleic acids. These are also impermeable because this number goes substantially low to less than 5 molecules per second. So, we say that these molecules cannot pass plasma membrane on their own. But does this mean that these molecules can never enter our cells? No, that would be wrong. Glucose can enter our cells. That's how we get nutrition. These ions enter and leave our cells. We study in physiology how nerve conduction takes place. So all these molecules, even amino acids, nucleic acids can enter and leave our cells. Alright, but they cannot do so on their own. They need certain type of transporter. They need some certain type of transporter proteins to get through this plasma membrane. That's what we will try to understand in today's lecture. Okay, so transport across plasma membrane can be readily classified into two types. One is passive transport and other one is active transport. So what do we mean by these transportation mechanisms? Before that, we need to understand what is concentration gradients and how diffusion takes place. For example, we have a plasma membrane here in the container. We have certain molecule which is permeable to this plasma membrane. The concentration of this molecule is way higher on one side of this membrane, and this concentration of the same molecule is lower on the other side. So, what will be the direction of transport? Because this membrane, we said, is permeable to this molecule, the molecule always go from an area of higher concentration to an area of lower concentration. This is the concept of simple diffusion. Okay, and if this molecule is permeable, this molecule can cross the plasma membrane. It do not require any kind of special transporter for it. Okay, now let's come to passive transport. What do we mean by passive transport? We mean two things by this word passive. The first is it follows concentration gradient, and when we say it follows concentration gradient, we are talking about this concept from an area of higher concentration to an area of lower concentration. This molecule goes from left to right. This is passive transport, point number 1. And for second point, this is passive. This does not require any energy to do so, because it can easily flow through this membrane from an area of higher to lower concentration, even thermodynamically. The free energy value will be negative in case. These are the two basic concepts of passive transport. Number 1, it occurs across concentration gradient, and number 2, it do not require any energy. Whereas in case of active transport, it occurs against the concentration gradient. The transfer of molecules or the movement of molecules will take place from an area of low concentration to an area of higher concentration. Alright, and to do this process we need energy in the form of ATP or any other energy source. Okay, these 2 points are still the same for active transport, but number 1, we need energy. And why do we need energy? Because we are acting against the concentration gradient. Okay, this was the basic nuance, basic difference between passive transport and active transport. We will study this active transport in a separate video, but for today we will be focusing on this passive transport. And in the beginning we said that this is the concept of diffusion, so we can classify passive transport into various types of diffusion. Alright, and for the sake of simplicity, we classify passive transport into either simple diffusion or a facilitated diffusion. And this classification is based whether this process of diffusion needs any transporter protein or not. If it does not need any special protein to transport, if this molecule is permeable through plasma membrane, like we said about gases like oxygen, this is known as simple diffusion. Alright, it does not require any special transporter protein. And if our molecule is like glucose, like fructose, like any specific amino acid, we need a specific transporter protein. And if we need a specific transporter protein, it is facilitated by the transporter protein, and diffusion is named as facilitated diffusion. Now, facilitated diffusion can be further classified on what kind of transporter protein is needed to carry out this diffusion process. Okay, so it can be classified into carrier mediated diffusion or channel mediated diffusion. We will see the difference between carrier mediated diffusion or channel mediated diffusion in a little while. But before that, we will discuss some points about simple diffusion. So we said simple diffusion is a type of passive transport. Passive transport means it does not need external energy. Thermodynamically, the value of delta G, the value of free energy, will be negative. So this process will be spontaneous, and this process occurs along the gradient from higher concentration to lower concentration. It occurs along the concentration gradient. So concentration gradient is directly proportional to diffusion. Okay, higher the concentration gradient, more will be the rate of diffusion. It is non-selective. Mostly it is non-selective, we can say that, and it shows non-saturable kinetics. What do we mean by non-saturable kinetics? That on a graph it will show a straight line. Okay, if it was saturable kinetics, it will show this S-shaped curve like we see in Michaelis-Menten kinetics. But it is non-saturable, so it will follow a straight line on this graph. Alright, we will study these graphs in a little while, but before that we need to know the first law of diffusion, or fix first law of diffusion, which states that the rate of diffusion is proportional to concentration gradient. Okay, so this is the basic formula for rate of diffusion. J here represents the flux of diffusion, and a negative sign represents that the process is spontaneous. The direction is from higher concentration to lower concentration. Okay, D is the diffusion constant. C or DC, the change in C, is concentration gradient, and dx is membrane thickness. Now, this diffusion coefficient depends on multiple factors. It depends on size of solute. If the size of solute is lower, the solute can easily diffuse. It depends on hydrophobicity of solute. As we know that plasma membrane is lipid soluble or hydrophobic in nature. If solute is hydrophobic, it will be easy for that solute to pass through this plasma membrane. It also depends on temperature and viscosity of the membrane. If membrane is highly viscous, the rate of diffusion may fall down, and if we increase the temperature, the rate of diffusion will also increase. So this is the Fick's law of diffusion, or we can say first law of diffusion. We talked about diffusion, that diffusion can be simple diffusion without any need of transporter protein, or if we need transporter protein, it is facilitated diffusion, and transporter protein may be a carrier or a channel. Let's talk in some details about these transporter proteins. What is the nature of these transporter proteins, and in what kind of transport these proteins are involved in. Okay, if let's start with this flow chart. If transporter protein is involved in a passive transport, we classify them into channel proteins. And if these transporter proteins are involved in either active or passive transport, we classify them into carrier proteins. So carrier proteins can do both active as well as passive transport, and channel proteins are only specific to passive transport. All right. So let's have a look at these channel proteins first, then we will come to these carrier proteins, and we will study these carrier proteins more in case of active transport. For this video we will be focusing more on these channel proteins. Channel proteins can be either be open all the times, or they can close or open with respect to certain stimulus. If they are open all the time, we can say that these are open channels, or these are leaky channels. And if these channel proteins open and close with respect to certain stimulus, we can classify these gated channels on the basis of this stimulus into three main categories. It can be voltage gated channels, it can be ligand gated channels, it can be mechanical gated channels. Okay, in case of voltage gated channels, it is the voltage or potential difference across the plasma membrane which determines whether this channel is open or closed. All right, for example, on the exoplasmic phase, if we have positive potential difference, then this channel is closed, and if we have negative potential on outside and positive on inside, for example, we have positive 20 millivolts inside and we have negative 40 millivolts outside, this channel opens, and in such conditions this channel can transport various ions. In case of ligand gated channels, there is a specific ligand. It may be some kind of protein, it may be some kind of hormone, or some kind of other chemical agent that binds to this channel in a specific manner, and when it binds to this channel opens and influx or efflux of certain molecule can take place. Okay, now this channel can have two conformations. This channel is having a transmembrane domain which is embedded in this membrane, and this channel can have outer exoplasmic domain or a cytoplasmic domain depending upon the conformation. If this channel is in such a position that it protrudes itself outside the cell, so these ligand molecules will be either proteins or hormones or certain kind of chemicals, and if this ligand binding domain is towards the cytosol, then this ligand will mostly be a nucleotide. All right, in case of mechanically gated channels, we have certain kind of mechanical stimulus which opens and closes these channels. Okay, now if we come to active transport based carrier proteins, they are classified on the basis of direction of transport. Okay, if transport is restricted to only one direction, we have unipolar. If it is it can transport in two directions, it is co-transporter. And co-transporters are further classified as symporter. If it transports both the molecules, if it transports both the molecules in the same direction, it is a symporter, and if it transports in opposite direction, it is antiporters, as you can see right here. So we talked about simple diffusion. Now let's come at facilitated diffusion. So what was the basic definition of this facilitated diffusion? That it will occur from an area of higher concentration to low concentration. This will not change, but it will require a specific transporter proteins. So it is a passive transport, it occurs along the gradient, it is having a selectivity, means we are involving some sort of transporters here. Transporter can be a channel or a carrier. If it is a channel, the specificity is very low, and if it is a carrier, the specificity is very high. For example, we have glucose transport. Now, if we have glucose transporter here, this will only transport glucose or hexoses. It can transport fructose or glucose or mannose, but it will not transport some other kind of amino acid. That's what we mean by specificity. Okay, we talked about this earlier, that simple diffusion shows non-saturable kinetics. It will be a straight line, but in case of facilitated diffusion, there is a certain limit on how many molecules can this transporter carry at a particular amount of time, and there are many such transporters on a membrane, and when these transporters are all occupied, they can no longer transport any more such molecules. So this shows a saturable kinetics. So this follows a curved shape on this graph. Now let's look at some examples of these transporters. Okay, for carriers we will be looking at glucose transporters, and for channels we will be looking at aquaporins. Let's start with glucose transporters. We will not see the exact mechanism here, that will be for a biochemistry or physiology class, but we will see the basic nature of transporter and basic process or basic fundamentals behind this glucose transportation. Glucose transporters are abbreviated as GLUT transporters. Alright, these are abbreviated as GLUT transporters. What is the nature of these transporters? So they are glycoproteins, they are monomeric proteins having a molecular weight of 45 to 55 kilodaltons, and the important thing to remember is they are 12 pass receptors. They span the plasma membrane 12 times, and the nature of these spans is alpha helix. So as you can see, this is 12 pass receptor, means even pass receptors, and even pass receptors are having a quality that they have N terminus and C terminus on the same side. Alright, and in the case of GLUT transporter, both N terminus and C terminus are cytosolic. Alright, if this is a membrane and this is glucose N terminus, this is first span, second, third, fourth, fifth, sixth, seventh, and so on. Here somewhere here comes C terminus. There are 12 such spans. Alright, and 1 2 3 4 5 6 7 8, this like, and this loop at 6th and 7th, this is susceptible for phosphorylation. This is important in many transportation mechanisms across this GLUT transporter. So this is somewhat important to know. Now this glucose transporter functionally is allowing facilitated diffusion, which is quite obvious by its name. This is glucose transporter. It can transport either glucose or various isoforms, which is specific for specific tissues. Now let's talk about glucose transporter classification. All we need to know that glucose transporters can be classified into 3 basic categories: the class 1, class 2, and class 3 transporters. Class 1 transporters are exclusive for glucose transport. They transport glucose across various cells. In case of class 2 transporters, they are exclusive fructose transporters. And in case of class 3, these transport some other molecules. These are specific for a physiology class, and we will need to know only a few of these transporters for now. For example, we have GLUT2 transporters which are present in liver cells, pancreatic cells, GIT cells, and all that. Okay, GLUT2 transporters show a bidirectional transport. They can transport glucose molecule inside the cell and outside the cell. Okay, so glucose 2 depends two direction, in and out of the cells. In case of pancreas, these cells senses the level of glucose. If the level of glucose is fairly high, okay, so these will undergo signaling and stimulate pancreas beta cells to release insulin, and insulin will bind to insulin receptors on some kind of cells like muscle cells and allow these cells to express another transporter named as GLUT4 transporter, which allows the excess of glucose to be entered into these cells to maintain the glucose levels. So for now we need to know is glucose transporter 2, glucose transporter 4, which is present in skeletal muscles, cardiac muscles, adipose tissue. And if we talk about fructose transporters, the most important one is GLUT5 transporter. It is present in small intestines, kidney, and testes. Why small intestines? It is directly responsible for uptake of fructose molecules we took from diet. The energy source for sperms is fructose in place of glucose. Okay, so that was all about glucose transport. And the transport process itself, how this transport takes place, we will talk in active transport lecture. For now, you can see that when the glucose is transported from GIT cells into blood supply, for example, this takes place through this GLUT transporter, which is showing the passive transport. But the situation here is completely different. Second most important carrier protein we will talk about today is chloride bicarbonate anion exchange. Sometimes we will see its name as band 3 protein. First of all, we start with what is the nature of this protein. Again, the nature of protein is a glycoprotein, and if it is a transporter, it has to be a multipass transmembrane protein. It is again 12 to 14 pass. This is even pass, and if it is even pass, N terminus and C terminus have to be on same side, and for this transporter and C termini both are on cytosolic side. All right. Functionally, it is a homodimer. Okay, here it is having two domains. One is cytoplasmic domain, other is membrane spanning domain. What is the function of this cytoplasmic domain? Its function is to anchor with cytoskeletal proteins. It anchors with ankyrin or spectrin. In case of membrane spanning domain, it is the domain responsible for transport of anions. And what are the anions this transporter is going to transport? These are bicarbonate ion and chloride ion, and this is transported in the fashion of antipode, both in the different direction. If the bicarbonate ion is going out, the chloride ion will be coming in. This is what we mean by antipoder. Just like we saw the classification of glucose transporter, here we will see classification of band 3 chloride bicarbonate ion transporter. It can be classified into AE1, AE2 and AE3 types. AE1 is what we described as band 3 transporter. It is present in red blood cells, it is present in kidneys, and it is the most abundant protein on the membrane of red blood cells. Alright, and all these other points you can go through on your own. In the process of breathing or gaseous exchange in the respiratory physiology, we will see the details of these transporting mechanisms of these the formation of these bicarbonate ions and transportation across the membrane of red blood cells. Here is our AE1 protein, which is band 3 protein we described here, and this is the antipode mechanism we can show in what we say systemic capillaries red blood cells, and in case of pulmonary capillary red blood cells the process flips over. Here bicarbonate is coming out here bicarbonate will go in. We will study the details of this process itself in the separate video of respiratory physiology. Now let's come to channels. Channels are also transmembrane proteins. They are multipass in nature. These two are similar to carrier proteins. They show passive transport, but in case of carrier proteins, transport can either be passive or active. And if it is passive specific in carrier proteins, it always has to be along the gradient. Sometimes we say downhill gradient because we are going from higher concentration to lower concentration. For carriers, they are less selective as compared to channels are less selective as compared to carriers, and they are mostly ion transporters. Mostly, not always. Okay, we will talk about two main type of channels in this video. First is aquaporins and second is ionophores. For aquaporins, these are multipass transmembrane protein, as you can see. Okay, these are 6-pass transmembrane proteins, 1, 2, 3, 4, 5 and 6. And if it is even pass, N terminus and C terminus has to be on one side, and here N terminus and C terminus both are on cytoplasmic side. And you can see there are different loops: loop A, loop B, C, D, E. Loop B is between span 2 and span 3, and loop E is between span 5 and 6. On both these loops, either it is loop B or loop E, we have specific amino acids. We have asparagine, proline and alanine, NPA domain. Okay, now this NPA domain forms the selectivity filter. Have you ever wondered how these aquaporins know what molecule to transport? This is due to this selectivity filter, NPA filter. We have amino acids, asparagine, we have proline, we have alanine, which is deciding the selectivity of this filter. They prevent passes of protons. Protons are small in size, they can easily leak out, so these specific amino acid prevents that from happening. They only allow the transport of uncharged water molecules, and they forms a barrier with electrostatic repulsion, and they also have some sort of size exclusion mechanism. So this was the importance of this NPA filter on loops B and loop E. You need to know. Now we also need to know what is the nature of this channel. Okay, this channel can either be leaky channel, this channel can either be a gated channel. Both types of forms are present. If it is gated, but will be the stimulus for this channel to open or close. The stimulus is change in pH, it can be change in calcium ion concentration, it can be change in concentration of reactive oxygen species, or it can be either by phosphorylation or dephosphorylation. The functional protein is homotetramer. This occurs in homotetrameric form, but in case of homotetramer, each of the monomer forms a channel. Okay, each of the monomer can act separately as a channel. Now, what is the classification of aquaporins? Aquaporins can be classified into 2 or 3 main categories. It can be either classical or orthodox aquaporins. Example is aquaporin 1 and 2. It can be aquaglyceroporins, which can transport glycerol, ammonia and some other molecules along with transport of water. The main example is aquaporin 3 and 7. And last one is unorthodox or non-traditional aquaporins. Example is aquaporin 11 and 12. Okay, we need to know only some of these aquaporins, like aquaporin 1 is present in PCT of kidney, some RBCs and choroid plexus. Most important is aquaporin 2 and aquaporin 4. The aquaporin 2 is present in collecting duct of kidney. Okay, it is also vasopressin regulated. It is regulated by ADH. And aquaporin 4 is present in brain or spinal cord. And if we talk about aquaglyceroporins, the main aquaglyceroporin we need to know is aquaglyceroporin 3, which is also present in kidney cells and responsible for transportation of reabsorbed water into blood supply, as you can see here. And in case of unorthodox aquaporins, we have aquaporin 0 in eye, and we have aquaporin 11 and 12 in kidney and pancreas. Let's see in brief this process of aquaporin 2, which is vasopressin ADH regulated. Vasopressin is present in blood supply. We are seeing these cells of collecting duct of a nephron, as you can see, collecting duct of nephron. ADH binds to ADH receptor, which is present on these cells, and through a certain kind of signaling, these cells have certain vesicles on which these aquaporin 2 are present in membrane of those vesicles, and these vesicles are made to fuse to these membranes. Okay, as you can see, this vesicle is fusing to this membrane, and another vesicle has already been fused, and this hormone vasopressin is water retention hormone. We are retaining water, we are reabsorbing water from the lumen from this aquaporin 2, and this water comes into this cell, and through aquaporin 3, which is aquaglyceroporin, and this water comes into blood supply, and here it can be utilized to regulate blood pressure. And this was aquaporins. Now last one for today's lecture is ionophores. Okay, these are different from any carrier or channel we have studied so far. Ionophorins are ion specific, but these are not the integral part of plasma membrane. These are lipid soluble and exogenous compounds. Okay, these are lipid soluble, exogenous, cyclic, and polyether compounds, highly selective for ions like potassium, sodium, or calcium, responsible for transport of ions along the gradient by shielding the charge of those ions. That is the main mechanism behind the functioning of these ionophores. Like any other transporter we studied so far, we will classify these ionophores into three main categories. They can be carrier forming ionophores, they can be channel forming ionophores, or they can be purely acting as antibiotics. Now these two can also act as antibiotics, but we have a separate category, carrier forming ionophores. They show reversible binding with ions, and they show passive diffusion, and this diffusion can be either electrogenic or electroneutral, depending upon what type of transporter it is and what type of ion it is transporting. The two main examples we need to remember in carrier forming ionophores is belinomycin and nonactin. Belinomycin is a potassium specific transporter, and it is mobile ion carrier. It is a cyclic peptide in itself, consisting of both D and L amino acids, specific for potassium transport, and it can act as antibiotic. Okay, in case of non-actin, it can transport potassium or sodium ions. We can also say this is alkali metal cation transporter as well. Okay, we are coming to channel forming ionophores. They form hydrophobic pore that can shield the charge and allow the ion to be transported across the plasma membrane. So it does this transport through electrochemical gradient, because if it is a charged ion, we are saying it is transporting along electrochemical gradient. Okay, two main examples are gramicidin A and amphotericin B. Gramicidin A is dimeric in nature, and it transports monovalent cations such as protons or sodium ions or potassium ions. Okay, in case of amphotericin B is fungicidal in nature because it binds to ergosterol, which is a type of sterol present in membrane of various fungi, and it binds to this ergosterol. It creates leaky pores, the efflux of potassium ions. And for the last category, we have ionophoric antibiotics, and these antibiotics disrupt homeostasis in microbes. The three main examples is monacin, nigericin, and calcimicin. Okay, in case of monacin, it transports sodium ions basically more than the potassium ions. It is showing anticoccidial activity of nigericin. It disrupts the pH gradient, and it is responsible for transport or exchange of protons and potassium ions. In case of calcimicin, as the name indicates, it is responsible for calcium transport. though it can transport magnesium as well, and it increases the cytoplasmic calcium concentration. Okay, so that was all about the passive transport across plasma membrane.